Unit 12 Bioprocess Engineering. This unit introduces as a core subject for GAT-B preparation, focusing on the principles and industrial applications of biological systems. It sets the foundation for understanding how biological processes are engineered and scaled to produce commercially valuable products. This emphasizes on building conceptual clarity that connects biology with real-world industrial processes. The title slide refers to the structural approach of this unit, which will systematically cover upstream processing, downstream processing, bioreactor systems, and industrial applications. As you progress through this unit, the goal is to develop both theoretical understanding and exam-oriented problem-solving skills relevant to bioprocess engineering. This slide highlights the structured preparation strategy required for excelling in GAT-B 2026, emphasizing concept-based learning combined with consistent practice. It focuses on the importance of understanding core concepts clearly rather than relying on rote memorization, which is crucial for solving analytical and application-based questions in the exam. Additionally, it outlines the resources provided, such as mock test, previous year questions, and doubt-solving support. These elements collectively ensures comprehensive preparation by covering the entire process from registration to counseling, helping students stay organized and focused throughout their preparation journey. Bioprocess engineering involves the integration of biology, chemical engineering, and processes engineered to convert biological materials into commercially useful products. This field plays a vital role in industries such as pharmaceuticals, food processing, agriculture, and environmental management. The objective is to design effective and scalable process that maximize productivity and maintain economic feasibility. The process is broadly divided into upstream and downstream processing. Upstream processing focuses on culture development and production, while downstream processing deals with product recovery and purification. Key goals include achieving high yield, productivity, and selectivity, while ensuring scalability for industrial applications. Upstream processing includes all the preparatory steps required for before actual production phase in a bioprocess. It begins with strain selection and improvement, which may involve mutation or genetic engineering to enhance productivity. This is followed by inoculum development, where seed cultures are scaled up to prepare for large-scale fermentation. Media formulation is another critical aspect, providing essential nutrients such as carbon and nitrogen sources required for microbial growth. Sterilization ensures contamination-free conditions, which is crucial for maintaining process efficiently. The overall objective of upstream processing is to maximize biomass or metabolic production under controlled conditions. Downstream processing involves the recovery and purification of the desired product after the fermentation process is complete. It includes sequential steps such as cell separation using centrifugation or filtration, followed by cell distribution if the product is intracellular. These steps are essential for isolating the target product from the complex fermentation mixture. Further, purification steps ensure that the product meets the required quality standards. Techniques such as chromatography, membrane filtration, and precipitation are commonly used. Downstream processing is often the most expensive part of the bioprocess, accounting for a significant portion of total production cost, making efficiency and optimization critical. Bioprocess design and scale-up involves transitioning a process from laboratory scale to pilot and eventually to full industrial production. This step is crucial because conditions optimized at small scale often behave differently when scaled up. Engineers must ensure that parameters such as mixing, oxygen transfer, and nutrient availability remain consistent across scales to maintain productivity and product quality. Key scale-up criteria include geometric similarity, oxygen transfer coefficient, and power input per unit volume. Challenges such as oxygen limitation, heat removal, and shear stress becomes more pronounced at large scales. To address these issues, scale-down models are often used for optimization, and all processes must comply with regulatory standards like Good Manufacturing Practices (GMP). Microbial cell culture platforms utilize microorganisms such as bacteria and yeast for large-scale production processes. Organisms like Escherichia coli and Saccharomyces cerevisiae are widely used due to their rapid growth rates, simple nutritional requirements, and high productivity. These systems are highly efficient and cost-effective, making them suitable for industrial applications. Microbial cultures are commonly used for the production of enzymes, antibiotics, and recombinant proteins. However, a major limitation is the lack of proper post-translational modifications in bacterial systems, which can affect protein functionality. Despite this, microbial systems remain the backbone of industrial biotechnology due to their scalability. Animal cell platforms involve the use of mammalian cell lines such as CHO and HER293 cells for producing complex biological products. These systems are essential when proper post-translational modifications are required, especially for therapeutic proteins like monoclonal antibodies and vaccines. This makes them highly valuable in the pharmaceutical industry. However, animal cell cultures have several limitations, including slower growth rates, higher media cost, and increased risk of contamination. They require more controlled conditions and sophisticated bioreactor systems. Despite these challenges, their ability to produce biologically active and structurally accurate proteins make them indisciplinary for advanced biotechnological applications. Plant cell culture platforms involve techniques such as callus culture and suspension culture to produce valuable secondary metabolites. These systems are particularly useful for synthesizing compounds like alkaloids and flavonoids, which have pharmaceutical and industrial importance. Plant-based systems provide an alternative to traditional extraction from whole plant. Advanced techniques like hairy root culture, induced using agrobacterium rhizogenes, enhance metabolic protection. However, plant cell cultures face challenges such as low yield and difficulties in scale up for industrial production. Despite these limitations, they are important for producing specialized compounds that are difficult to synthesize chemically. Biomass production refers to the generation of cellular materials during microbial growth and is often associated with growth-related processes. Primary metabolites are produced during the exponential phase of growth and include essential components such as ethanol and amino acids. These metabolites are directly linked to cellular metabolism and growth. In contrast, secondary metabolites are produced during the stationary phase and are not directly involved in growth. These include antibiotics and pigments, which have significant industrial applications. Understanding the kinetics of growth, including parameters like growth rate and substrate utilization, is essential for optimizing production and maximizing yield in bioprocess systems. Industrial bioproducts are derived from biological processes and plays a crucial role in sustainable development. These include biofuels such as ethanol, biodiesel, and biogas, which serves as renewable alternatives to fossil fuels. Bioplastics like PHB are biodegradable and environmentally friendly, reducing plastic pollution and dependence on petrochemical resources. In addition to fuels and plastics, industrial enzymes such as amylases, proteases, and lipases are widely used in food, detergent, and textile industry. Recombinant protein protection involves the expression of foreign genes in suitable host systems such as bacteria, yeast, insect cells, or mammalian cells. The process begins with gene cloning, followed by transformation into a host organism and subsequent expression and harvesting of desired protein. This technology is widely used in production of therapeutic proteins and industrial enzymes. Vectors such as plasmids and viral systems are used to introduce the gene of interest, and expression is often controlled using induction systems like IPTG or temperature shifts. However, challenges such as protein misfolding and formation of inclusion bodies can affect yield and functionality. A well-known example is the production of human insulin using recombinant DNA technology. Chromatographic techniques are essential in downstream processing for the purification of biomolecules based on their psychochemical properties. The principle involves differential partition of components between a stationary phase and a mobile phase. Different types of chromatography are widely used depending on the property being exploited, such as charge, size, or binding affinity. Ion exchange chromatography separates molecules based on charge, while size exclusion chromatography separates them based on molecular size. Affinity chromatography provides high specificity using a ligand target interaction, and hydrophobic interaction chromatography separates based on hydrophobicity. Key parameters such as resolution, selectivity, and capacity factor determine the effective of separation, although these methods can be expensive. Membrane-based separation techniques operate on the principle of selective permeability, allowing certain molecules to pass through while retaining others. These methods include microfiltration, ultrafiltration, nanofiltration, and reverse osmosis, each defined by its pore size and separation capability. They are widely used for applications such as cell harvesting, protein concentration, and water purification. The driving force for membrane processes typically a pressure gradient, making them energy efficient and scalable. However, major limitation is membrane fouling, where particles accumulate on the membrane surface and reduce efficiency over time. Despite this, membrane technologies are essential component of modern downstream processing due to their versatility and effectivity. Protein purification is a multi-step process designed to isolate the target protein while high purity and yield. It typically involves three stages: capture, intermediate purification, and polishing. Each stage progressively removes impurities and enhances the quality of the final product, ensuring it meets industrial and regulatory standards. Various techniques such as ammonium sulfate precipitation and multiple chromatographic steps are employed during purification. Key performance metrics include yield, purity, specific activity, and fold purification. A critical concept in protein purification is the trade-off between purity and recovery, as achieved high purity often results in low overall yield. Immobilization of biocatalysts refers to the confinement of enzymes or whole cells within a defined space while retaining their catalytic activity. This technique allows the biocatalyst to be reused multiple times, improves process efficiently and reducing overall production cost. Common immobilization methods include adsorption, covalent binding, and encapsulation, each differing in stability and strength of interaction. Various support materials such as alginate, silica, and synthetic polymers are used to immobilize the biocatalyst. The major advantage includes enhanced stability, ease of separation, and suitable for continuous processing. However, limitations such as diffusion resistance and potential loss of enzymatic activity must be considered when designing immobilized systems. Enzyme immobilization has significant industrial applications due to its ability to improve operational stability and allow repeated use of enzymes. For example, glucose isomers is immobilized for the production of high fructose corn syrup, and lactase is used in immobilized form for producing lactose-free milk. These applications demonstrate the economic and functional advantages of immobilized enzyme systems. Immobilized enzymes are commonly used in reactor systems such as packed bed and fluidized bed reactors. The kinetics of immobilized enzymes often derive from classical Michelmann behavior due to massive transfer limitations. A key challenge in these systems is balancing intrinsic reactions rate with diffusion resistance to achieve optimal performance. Cell immobilization involves the use of whole cells contained within a matrix, allowing them to perform complex biochemical reactions. This approach is particularly useful for multi-step processes where multiple enzymes and cofactors are required. As the entire metabolic pathway remains intact within the cell, it also enables cofactor regeneration, which is advantageous for continuous bioprocesses. Common methods include gel entrapment using materials like alginate and the formation of biofilms on surfaces. These systems are widely applied in wastewater treatment and fermentation processes. The advantage includes improved robustness and long-term stability, although challenges such as mass transfer limitation and reduced cell viability over time may arise. Bioremediation is the use of microorganisms to degrade or detoxify environmental pollutants, making it an important application of bioprocess engineering in environmental management. It can be carried out in situ where treatment occurs at the site of contamination, or ex situ where contaminated material is removed and treated elsewhere. This approach is efficient for pollutants such as hydrocarbons, heavy metals, and pesticides. Microorganisms like Pseudomonas petita played a vital role in metabolic versatility. The efficiency of bioremediation depends on several factors, including nutrient availability, oxygen levels, pH, and temperature. Optimizing these conditions is essential to enhance microbial activity and achieve effective pollutant degradation. Aerobic bioremediation process involves the degradation of pollutants in the presence of oxygen, which act as the terminal electron acceptor. These processes convert organic contaminants into simpler end products such as carbon dioxide and water through oxidative metabolic pathways. Techniques such as bioventing, biospacing, and composting are commonly used to supply oxygen and enhance microbial activity. Aerobic processes are generally faster and more efficient compared to anaerobic methods, making them suitable for many environmental applications. However, oxygen transfer can become a limiting factor, especially in soil systems where diffusion is restricted. Proper aeration strategies are therefore essential to maintain optimal degradation rates and ensure effective remediation. Anaerobic bioremediation involves the degradation of pollutants in the absence of oxygen, where microorganisms utilize alternative electron acceptors such as nitrate, sulfate, or carbon dioxide. These processes result in the formation of end products like methane and carbon dioxide, making them particularly useful in energy generation systems such as biogas production. Methanogenic microorganisms such as Methanobacterium plays a crucial role in these pathways by converting intermediate products into methane. Anaerobic processes are widely applied in sludge digestion and landfill treatment due to their ability to handle high organic loads. Additionally, they offer the advantage of energy recovery, although the degradation rate is generally slow compared to aerobic systems. Waste stabilization focus on reducing the toxicity, volume, and environmental impact of both solid and liquid waste through biological treatment process. Techniques such as the activated sludge process, trickling filters, and anaerobic digesters are commonly used to treat industrial and municipal waste streams. These methods rely on microbial activity to break down organic matter into simpler and less harmful substances. Key parameters used to evaluate the efficiency of waste treatment includes biochemical oxygen demand (BOD) and chemical oxygen demand (COD), which indicate the level of organic pollutants. Effective waste stabilization is essential for maintaining environmental safety and is widely implemented in effluent treatment plants across various industries. This slide provides a comprehensive revision of key concepts in bioprocess engineering, summarizing the entire workflow from upstream to downstream processing. It reinforces the idea that bioprocess engineering integrates biological systems with engineering principles to enable effective industrial production. Important components include strain development, fermentation control, and product recovery. It also highlights critical exam focused areas such as the distinction between primary and secondary metabolites, the role of oxygen transfer, and the difference between batch and fed batch process. Additionally, it provides essential downstream techniques like chromatography and membrane filtration, along with applications such as bioremediation, ensuring a complete conceptual overview for revision. Subscribe BioBrains and stay updated with the latest GAT-B 2026 preparation tips.